So if you have any, if there are any issues with the PowerPoint, I do have the chat open. So if the slides don't seem like they are moving properly, just send me a message, okay? And I'll see if I can fix that. All right. So just a reminder before we start with today's lecture that you do have a homework assignment that's due this Friday night by midnight, or I think technically like 11:55 or whatever, however Laulima sets it up. So don't forget to do that. All right. All right. So let's continue talking about the different changes and challenges that a cancer cell faces as it leaves the original primary tumor and has access to invade the surrounding tissues and eventually get into the blood. So we discussed the importance of epithelial to mesenchymal transition as part of this, and we'll take a closer look at the different factors that govern how well different cells that are circulating in the blood, cancer cells that come from the original tumor, how well they can actually disseminate to form secondary metastatic lesions. Let's review some key concepts. So remember that primary tumor cells are encouraged by reactive stroma in order to invade and intravasate nearby capillaries. Remember, this reactive stroma is the confused and sort of manipulated stroma that up until this point had been sort of, just kind of supporting the epithelial cells that were growing. But now that the tumor has reached a particular size and is facing the challenges of being too big, right, the tumor needs the stroma that's surrounding it to start behaving differently and behave in a way that actually gives that tumor some sort of emergency resources for that tumor to be able to continue growing out of control. Right? And so we say the stroma becomes reactive. Part of this is that the tumor learns through different stumbling upon mutations to recruit macrophages that remodel the surrounding tissue and importantly remodel the extracellular matrix. Right? And by dissolving away things like the basement membrane, then this helps facilitate or encourage the carcinoma cells to undergo epithelial to mesenchymal transition, or EMT. This transition from a more epithelial state to a more mesenchymal state is marked by certain changes in protein expression, including things like downregulation of cell-to-cell adhesion molecules like E-cadherins that normally epithelial cells would rely upon to stay put in the right spot with their neighboring epithelial cells and only be happy, right, if they are properly in the right spot. Right? So if you lose E-cadherin expression and instead mistakenly start upregulating other kinds of cadherins, then encourage more mesenchymal cell interactions that would encourage the epithelial cell to be okay with leaving the original tumor site and making its way through the surrounding reactive stroma. We also learned that in order for the disseminated tumor cells, or the DTCs, that have been scattered all throughout the body, stuck in various tissues and organs, in order for them to really go beyond just dividing a couple times and forming little micrometastases in those new locations, if they are to become large secondary tumors, they have to revert back to their epithelial state. Right? So we say that the reverse of EMT is MET, or mesenchymal epithelial transition. Right? And at that point, then you've sort of completed this whole metastases cascade, the steps that have now led to a large, life-threatening secondary tumor. And we said that the vast majority of deaths from cancers, from solid tumors, are a result of these metastatic lesions. Why? If you think about it, right? The primary lesion, the original primary tumor, has been growing in that particular spot in the original tumor, in the original tissue of origin for many years, right? So if that has been tolerated, that implies that that original location of the tumor, right, is still compatible with normal tissue function, throughout the body. Right? But once you have these large tumors forming as a result of metastases, right, that's very disruptive, right? And those disrupted tissues all throughout the body, right, the failure or the compromise of those is what leads to death. All right, so consider how much of this metastatic phenotype of a large aggressive tumor is really due to the tumor cell's genotype itself versus the encouragement of surrounding tissues. In other words, if we think about and revisit this classic like seed and soil model of metastases, how much of the success of a flowering plant growing from its seed is the result of the seed itself and the genome of the plant, and how much of it has to do with the right combination that's provided by the soil, right? I mean, I think you can understand that it's really both, right? The genotype of the plant seed has to be the right type to be okay in a particular soil, and that soil that it's in, right? What makes it the right type is that it has certain types of qualities, characteristics, moisture, nutrients, whatever, right, to support that seed. So think about it that way in terms of metastatic cancer cells as well. Let's take a closer look at some examples of how cancer cells talk with the surrounding stroma, right? This is this idea of dialogue now, right? We've gotten away from just thinking about an individual cancer cell as being a messed up phenotype on its own, right? That it's spent years reaching this point where its genome is messed up in, unfortunately, a combination that continues to allow it to become, right, an aggressive tumor. That was monologue. The last couple lectures we talked about dialogue, how the surrounding stroma plays a very important part of that, and signals go back and forth between the cancer cell and the stromal cells. So this cartoon illustrates that very nicely. The relationship between tumor cells and macrophages, right? These immune cells that are normally part of the stroma, they are part of the heterotypic cellular environment of the stroma. The macrophages that can be fooled into actually helping the cancer cells. So in this case, there's a particular signaling molecule known as colony stimulating factor, or CSF1. So this is a very important factor for mobilizing macrophages, getting them to fully differentiate from monocyte form into macrophage form. And it's also very important for activating them. On the same time, there's another important factor, the EGF, the epidermal growth factor that we sort of examined throughout the semester when we talked about things that signal through receptor tyrosine kinases and other growth factor type of receptors. Right? So EGF stimulates cancer cell growth and invasion. Right? So you have one factor, the CSF, that's talking to macrophages and encouraging macrophages. You have another factor, this EGF, that if it's received by the cancer cell, will stimulate additional aggressive cancer cell activity, right? So this is now a loop. So let's look here in this cartoon. As this green invasive carcinoma breaks free through the basement membrane, right, and enters the stroma, it has stumbled upon mutations that is causing it to express CSF1, colony stimulating factor. What does that do? That tends to encourage more macrophages to come and actually associate with this cancer cell. So here you have macrophages that were already in the tissue. So these are tumor-associated macrophages, or TAMs, right? So you can see some of them are actually coming out from the blood supply itself, undergoing diapedesis, squeezing out of the capillary, right, being stimulated by this CSF1 to migrate and come here and fully activate and become a fully differentiated macrophage. Right. So in return, the macrophages produce EGF, right, to try to stimulate the growth of epidermal cells. Why? Remember, this is all part of that sort of wound healing process, right? Macrophages that come to the site of a wound are there to do two things, right? They're there to try to eat up any bacteria or other pathogens that might have entered from the wound. And importantly, they are engaged in remodeling the damaged tissue so that new tissue can grow and have a place to grow, right? So EGF is part of how these macrophages would stimulate other epithelial cells to grow and mobilize. You can see also the macrophages assist in opening up the capillary even more by grabbing onto endothelial cells and sort of prying them away so that you have much more flow of these different healing type of factors, like platelets, for example, right, that can get to the site here. So let's look on the right. Here is some Western blot data. Remember, Western blots are when you actually run proteins on a gel, blot them, and then use specific antibodies to try to see the presence of these different proteins of interest. So they've taken cell lysate, so a whole bunch of macrophages and a whole bunch of carcinomas that they then lysed and taken all of the nice protein guts out of them, right? And then they've run these gels and they're doing Westerns on those two samples. So one of the things that oftentimes on a Western you have to probe for is some sort of a reference protein. So for example, beta-actin, which is just a normal part of the cytoskeleton, right? All cells should have beta-actin more or less in the same amounts. And so that's how you can make sure that the intensity of brightness of the beta-actin in the macrophages being similar to the intensity of brightness in the carcinoma cell lysate lets you know that you're loading, you know, comparable amounts or equal amounts of total cell lysate into two lanes, right? So it's not like somehow your results are biased just because you unwittingly loaded, you know, 1/50th the amount of macrophage lysate compared to, like, you know, the carcinoma cells, and then you're trying to compare the brightness of bands of other proteins, you'd be thrown off, right? So the beta-actin shows us, okay, good, right? Similar amounts of total protein are being loaded here. Then you can look for specific proteins within that. You can see that the carcinoma cells express EGF receptor on their surface, right? Because they're ready and willing to receive EGF signal. Macrophages don't, right? Macrophages don't care about EGF receptor, but the macrophages make the EGF itself, right? That's why you can see the macrophages make EGF. The carcinoma cells don't make EGF, right? So this is one direction of communication. Macrophages making EGF that are then received by the carcinoma cells' EGF receptors, right? And the flip side of this is that the macrophages, they make CSF1 receptor proteins on their surface because they would love to receive CSF1, right? And it's the carcinoma cells in this case that have stumbled upon, right, mutations that allow them to make a lot of CSF1, right? So that's why in this picture here, these are the little back and forth dialogue messages that are taking place. Not surprisingly, if you eliminate CSF1 signaling, then you can really dampen or discourage the aggressive spread of invasive carcinoma cells. So here in the lab, they've engineered some mice to be CSF knockouts, right? So these mice, the cells no longer produce any CSF at all. As a result, the tumors that are forming, right? So here are the wild type tumors that these carcinoma cells can still make CSF. Right? You can see very clearly that there's a lot of invasion. So this is the original tumor carcinoma cells up here, maybe down here there are some as well. And you can see these dark purple ones, they begin to really spill into the surrounding stroma. Right? So they'd be invading the stroma here, probably because a big part of it is they've learned how to make CSF, right? And that's attracting macrophages to help them out. In those CSF knockout mice, you can see a very different story, right? Those carcinoma cells are still very much contained in situ, right? Surrounded by their basement membrane, because they don't know or they cannot make functional CSF signals to attract macrophages to help them out. Right? Another such type of signaling molecule is hepatocyte growth factor, or HGF. Keep in mind that even though it's called hepatocyte growth factor, what's a hepatocyte? Right? Hepatocyte is a liver cell. Many times, I think we've talked about this, the naming of different proteins is kind of outdated, right? Or it's a little bit, we've moved on for that. Originally, hepatocyte growth factor was identified in liver cells. So the researchers who first identified it, just called it, a liver-specific, growth factor, right? Hepatocyte growth factor. Now, of course, we realize many years later that HGF is actually used by many different types of cells in the body, not just liver cells, but the name, still sticks. Right? So HGF stimulates invasion. Here is a neat way of looking at tumor invasion, both on a traditional monolayer type of cell culture experiment. So here you have some cells. Right? These are cells. I don't actually remember what type of cell these are. For argument's sake, we can just say these are normal hepatocyte cells or liver cells. Right? And on plastic, they would grow in a nice, well-behaved monolayer. Right? And they would stay together, because these epithelial cells, right, they want to have good communications with each other, right, in order to know that they are in the right place and they are also fixed on the basement membrane. Of course, in this case, the basement membrane is the plastic surface of the tissue culture dish that's been treated with certain types of coatings, different types of coatings that kind of simulate the basement membrane, and that's why epithelial cells will happily grow on it. Right, but the epithelial cells remain clustered and they remain, very well behaved. What you can do that's becoming more and more common is, one limitation of growing cells on plastic in a two-dimensional surface is that doesn't really represent the three-dimensional environment of the tissues of your body, right? So another way to culture these cells is to grow them in a three-dimensional collagen matrix. So in a gel, right, where you can go ahead and seed cells throughout the gel in three-dimensional space, right? And these cells, as they're dividing, right, they're dividing in 3 dimensions. So you get these little foci, or little pockets of cells growing. But again, very well behaved, and they stay together because they like that cell-to-cell adhesion. But if you go ahead, so this is in normal epithelium, normal cell media in the flask on the left. If you give them media that's highly, highly enriched, it's like spiked with high levels of HGF in it, this is what happens, right? In the tissue culture plate, the cells begin to separate from their neighbors and spread out. In three-dimensional space, you can really see them beginning to spread out, right? And this is like mimicking the invasion of surrounding tissues that advanced tumors would display. Part of the migration has to do very much with the ability of things like epithelial cells to be happy, no longer being epithelial-like in behavior, right? Remember, both having to do with being okay with not being attached to each other and being okay with not being attached to the basement membrane. We said that during wound healing, right, there are certain events that must happen that get epithelial cells mobilized in order for them, like the cells, the healthy cells that are on either side of a wound, like a cut in your skin, right, those epithelial cells have to undergo EMT to become more mesenchymal so that they're okay with detaching from where they normally are, migrating into the site of the wound, right, and then beginning to grow to form new healthy tissue. There are certain transcription factors inside of cells that encourage EMT. So these are what we just simply call a whole group of EMT transcription factors, or TFs. Not surprisingly, it's easy to forget, right, that all of these come from genes, human genes, and all of the cells in our body have these same genes in them, right? So why is it that all of the cells in our body don't go crazy and start moving around all over the place? It's because why? These genes in the majority of cells in our body have been more or less permanently shut down, right? Through things like, Well, hypermethylation, right, of promoters. So let's look at some examples of these. So 3 very important EMT transcription factors are snail, slug, and twist, right? Kind of funny names, right? Clearly, these were not originally identified in people. Right? I actually don't know the etymology or why snail is called snail and slug is called slug. I keep meaning to like look it up, but I always forget, right? But I like it because it's kind of humorous, right? You can see it was first identified in Drosophila. A number of these were first identified in Drosophila. Maybe their names have to do with some sort of weird phenotype that arises if they're mutated. But what these do is they help encourage the motility or the movement of epithelial cells, right? Therefore, encouraging EMT. You can see here that these genes are very actively expressed during embryonic development. Right? So many of these EMT transcription factors are highly active and expressed early in embryonic development. Why? Well, hopefully intuitively, you could figure out it's because early in development, when you have many cells dividing, right, in order to form the sort of rudimentary or beginning hints of different structures of the body, right, including things like the layout of the body, different types of compartments, maybe things like limbs, these epithelial cells, right, have to move around a lot. Right? So here in a cross-section of a Amphioxus embryo, so Amphioxus is it's an invertebrate. I think it's like a lancet, right? A very small invertebrate. But in cross-section, you can see at this point in development, right, these cells on either side of this sort of inner layer of embryonic cells are expressing high levels of snail, right? So those cells are kind of moving around, changing their shape as part of forming the neural crest of the creature here, right? Here in Xenopus, so frog embryo, you can see at this point, slug protein expression is really ramped up in these particular cells that are in this part of the embryo, right? At this point, I mean, the embryo is maybe like 1 million or 2 million cells or so, right? But very clearly, certain cells are expressing a lot of slug, right? And you can see it's beginning to form, again, the neural crest, right, of the embryo. And here, twist. So this is in fly Drosophila, right? At this point of the embryonic development, look, when the fly larva is nothing more than just basically like a ball of cells, right? These cells on the top begin to express high levels of twist, and that allows them, this is a nice illustration, to really start moving, right? And they're going to now start moving in here to start forming these different layers of the mesoderm. Right? So this is true in human embryos as well. Right? As part of early embryonic development, different cells, as part of the developing embryo, express these transcription factors so that the embryo has its proper shape and morphology. But as we develop, right, into a fetus, and then eventually we reach full term and we're born, right, the cells in our body, right, turn off the expression of snail, slug, and twist because it's not needed anymore. Right? But is that really true? Right? Because we talked about wound healing and how under the conditions of wound healing, you actually need to have epithelial cells suddenly be okay, becoming mesenchymal. Right? So not surprisingly, during wound healing, these EMT transcription factor genes get turned on again, right? Only in those cells that are right at the site of the wound in order to get those cells to participate in moving around and trying to heal the damaged tissue. So in adult bodies, right, these EMT transcription factors, like snail, slug, and twist, right, get transiently re-expressed. Right? The genes get reactivated, and you can see here. So this is in the lab. Again, remember I told you how people who study wound healing, they'll often grow a monolayer of epithelial cells on plastic in a dish, and then they'll just physically scrape sort of like or carve a channel through them, and then study what happens, right, to the cells on either side of that channel. So you can see after 24 hours of simulating a wound, right, where the cells on the bottom were scraped away, the cells at the boundary of the wound are expressing slug protein again, right? And that is encouraging the cells to migrate. After 48 hours, you can really see that the cells have begun to migrate to fill in there. At a certain point, right, once enough of this is filled in, the cells begin to turn off expression of these EMT transcription factors again, right? And after 96 hours, right, you see something like this. Right now, obviously, you're like, well, wait a minute, this is still like part of the simulated wound here. But yeah, but obviously this is done on a plastic surface in a dish, right? So you're not going to really see all of the wound healing things going on because you don't even have here other types of cells helping out, right? This is just looking at how within this time, these epithelial cells are undergoing EMT to become more mesenchymal because they are turning on again, right? Things like snail, slug, and twist. And at a certain point, they have to then reverse this and become epithelial again. Right? So snail, slug, and twist repress the expression of certain genes like the CDH1 gene, and the CDH1 gene is the gene that codes for E-cadherins, right? So this is helping them give up on their dependence on being stuck to a fellow epithelial cell. Remember, EMT transcription factor genes are normally active in the early embryo, but silenced in our adult cells, except for these brief episodes of wound healing. Right? And so that's another reason why the wound healing response, right, contributes to these types of EMT changes, because not surprisingly, right, the wound healing response will stimulate this, but what if, even before the tumor has gotten to the point of needing that wound healing response, what if the regulation of genes like snail, slug, and twist have become sort of part, right, of the random mutations that are taking place inside, right? In other words, what if the regulation or the silencing of these genes has been reversed just through mutations? Right? And you have tumor cells that are actively expressing snail, slug, or twist all the time. Right? So we know that when you expose cells to these EMT transcription factors, right, it makes them more stem cell-like. So for example, here are some human mammary epithelial cells, these HMLEs. And if they're normally growing and behaving well, right, they stick together because they're epithelial cells and they're stuck to the surface of the plastic, which is their basement membrane. If you were to do some flow cytometry and actually look at different markers expressed by these cells, you can see that you can divide this up here. If you're looking at markers CD44 and CD28 on the surface of these cells, cells that express high levels of CD44 tend to be cells that are more stem cell-like. Right? Cells that express high levels of CD28 tend to be cells that are more differentiated or less stem cell-like. Right? So in the healthy, normal state of these cells, right, if you go ahead and stain for these two, and then you look at where these individual cells sort of fall on the expression levels of CD24 and CD44, what you see is the vast majority of these normal epithelial cells would be in this more non-stem cell partition, right? However, if you expose them to a lot of snail transcription factor, you know, when I say expose them, I mean most likely you transfect them or make them start expressing snail inside of them, you can see a dramatic shift, right? This population of epithelial cells now shifts over to becoming more stem cell-like. And that's true if you get them to overexpress twist, right? They become more stem cell-like. Right? So part of being more stem cell-like, as you can see from the images up here, is they no longer have this dependence on being stuck to each other, and to a certain degree, less dependence on always being fixed on the basement membrane. And importantly, as you shift from being more of a differentiated cell to becoming more of a stem cell, right, you shift back. You shift from the focus of being a cell doing its normal differentiated functions back to a stem cell, which is more concerned with proliferating, right? So these things will then, in different ways, contribute to the increased aggressiveness of the tumor. So you can see here, here's an experiment looking at how EMTs can induce stem cell behavior in cells that are growing in more of an in vivo environment. So this is a stromal fat pad. So this is some tissue taken from a mammary, right? Mammary tissue. And so it provides a nice sort of stromal environment that you can now do some experiments and put different epithelial cells into. So they've taken away the ducts, for example, that exist in the stroma. And then they go ahead and they implant some test cells in here and see, can these cells go ahead and grow into, a new set of ducts, right? So you can see here that if you have control epithelial cells, if you just stick them into the stroma, right, and you don't give them any other kind of encouragement, right, these epithelial cells are just sitting here in this fat stromal pad, right? They might not do anything because these are normal, healthy cells that are kind of like, I'm not going to worry about proliferating, right? They'll just sit there being a normal differentiated cell. But if you go ahead and you douse them, right? So this is 10 to the 3 or 1,000 control MECs, these memory epithelial cells, right? And you see hardly any growth, right? Here you take a tenth of them, 100 of these MECs that have been exposed to different types of EMT transcription factors. So in addition to a slug, here you are also exposing them to SOX9, right? Another EMT factor. And look at the dramatic difference, right? These 100 MEC cells, right, start dividing and proliferating and reconstituting the ductal formation of these different glands in the tissue here. So, oftentimes throughout the semester, when we learn about a new transcription factor or a new signaling pathway, I show you some clinical data to remind you that all of the stuff that we learn in the class, it is relevant, right, to what cancer patients are evaluated on in clinics today. So here's the clinical significance of EMT transcription factors in prognosis. So you can see we're looking at 3 different types of cancer here: breast cancer, melanomas, and gastric cancers. In each one of these cases, these are Kaplan-Meier plots looking at if you have a group or a cohort of patients who have breast cancer, for example, and they first present in the clinic and biopsies are taken, right? The pathologist will look for all kinds of things. If it's breast cancer, they'll look for things like HER2/neu, they'll look for MYC, right? And they might look for different types of EMT factors like snail. So you can see here, these 2 groups, the patients who presented with tumors that had low evidence of snail expression are in green, compared to patients whose tumors at the first time when they presented in the clinic already showed high levels of snail expression, right? Just from random mutations reactivating the expression of snail, right? And you can see what a night and day difference, right, between the prognosis. So on the y-axis is percent of response or relapse-free survival, right, followed over several years. So this holds true in melanoma as well, right? Low twist expression in the tumors at the time that the patient was first treated versus high twist, right? In gastric cancer, right? Tumors that had absence of slug expression versus tumors that started off with high levels of slug expression, right? So these are very important diagnostic tools for clinicians, oncologists to know about, to be able to help map out or plan a course of aggressive treatment for a patient, depending upon things like the status of these kinds of transcription factor expression levels. Okay. Another example of how cancer cells, carcinoma cells, can work towards remodeling their stromal environment. Up until now, I think I sort of made it sound a little incorrectly, as though somehow the carcinoma cells are entirely dependent upon other cells in the stroma to get fooled or tricked, right, into sort of initiating a wound healing response. And that's how macrophages end up digesting away the basement membrane and the surrounding tissue, making it easier for the cancer cells to invade. But that's not entirely true, because, there are many genes inside of the cancer cell itself that, if they become accidentally reactivated or even activated in the first place where they shouldn't be, right? So many genes, obviously, an epithelial cell has no business even expressing in the first place, right? That can actually cause or contribute to the cancer cell itself having newfound properties to be able to invade better. So the expression of matrix metalloproteinases, right? These are those proteases that are normally expressed by macrophages to digest away the ECM, right? What if the cancer cell itself, right, improperly reactivates or turns on the expression of certain proteases like this? So here's an example. This is kind of an interesting one, right? This is a membrane type 1 MMP, right, or MT1 MMP. So when it is expressed, so it's a protease also that will digest other proteins, but when it is expressed improperly by a cancer cell, it remains on the cancer cell itself. So it's a membrane-bound protease, right? This enables the cancer cell to itself digest away the surrounding stroma. So you can see here, right? So that's what these little red Pac-Man are, right? These little MT1 MMPs on the surface of this cancer cell. And it's just kind of munching away, carving itself through the stroma, right? So it carves like a little channel here to make it much easier for other cancer cells to spread into as well. Not only that, but of course, it also chews away the stroma to allow for things like angiogenesis to happen. So here's a tumor with wild type MT1 MMP gene that has been now improperly activated. This is amazing, right? So this is very clearly a three-dimensional culture that we're talking about here, right? So this is a tumor that's growing, and the cells are now expanding out into the surrounding three-dimensional space, the stroma, right? And you can see that here. So that's if these cells have wild type MT1 MMP. In a tumor with knocked down MT1 MMP, you can see, right, it is highly impaired from being able to spread, right, into the surrounding environment. This is a super cool image because these are fibrosarcoma cells that have learned to activate MMT1 on their surface. And what you see here is the pink represent the cancer cells. Now, you obviously cannot see the MMT1 MMP on their surface because, we're just talking about small little proteins on here. But the blue are the collagen fibers of the extracellular matrix, right? So when you look at this, you can really imagine, right, these cancer cells carving away at the surrounding extracellular matrix as part of their invasion. All right, so some important take-home messages from this discussion. One is that the heterotypic signals from stroma, meaning cells that are coming from different cell types in the stroma, are very important for invasion. Right? And it's the buildup of mutations and changes inside the cancer cell itself are not enough. Right? Monologue is not in itself enough. The cancer cell can yell and scream as loud as it wants, but as a monologue, right, there is no response to help out. You need a dialogue in order to get invasion to happen. Importantly, another key thing we talked about is EMT does not need to wait for a whole bunch of random mutations to happen over time, because certain master programs of gene expression are ready to go at a moment's notice. And we evolved that way, right? Because when you get a cut in your arm and you're bleeding, you cannot wait hours right, for the cells there to sort of, oh, start turning on different genes, right? Because meanwhile, you're just bleeding there for several hours, right? That's not good. We evolved so that certain sets or certain programs of genetic pathways can be all turned on at once, and that is the wound healing response. So part of that is the reactivation of these EMT transcription factors, right, that should normally be silent in these cells, these differentiated cells, but they can be transiently reactivated to get the cells to move as part of healing. Right? We also said, of course, it's very possible that some of these EMT genes have already become improperly activated and switched on in the tumor cell itself due to random mutations, right? So you bet those types of cells would also be very aggressive in nature. And point three, because invasion is driven by the reactive stroma, this confused environment, right, at the primary tumor site, once you have a motile cancer cell that has entered the blood and gotten somewhere else in the body, somewhere else where there is normal stroma and not reactive stroma, right, that cancer cell is now removed from that kind of very crazy environment of the reactive stroma, right? It wants to sort of resume its normal epithelial-like properties, including reattaching to the basement membrane and sort of establishing a new relationship with the healthy stroma that's surrounding it, right? Causing that stroma, of course, to be dramatically altered in the process. So for the last couple slides, let's just look at some examples of why there are different metastatic tendencies to different types of secondary tumor tissue sites. Right? So as we learned, when you have disseminated or circulating tumor cells that have made it into the blood, they can potentially circulate anywhere in the body because they're just riding the blood, right, and randomly going left and right as large blood vessels fork into smaller ones, which fork into smaller ones still, eventually ending up, right, being caught in tissues all throughout the body. We said that only some tissue environments would be the right soil combination for that seed to be happy in, right? And that combination then depends upon the genotype of the seed, in other words, the genotype of the cancer cell itself and its own unique, weird, messed up properties. And it depends upon the nature of the soil or the new healthy stroma that the disseminated tumor cell finds itself in. So because of that, there are only several places that you might expect metastases to commonly occur. Right? And that's not the same depending upon different cancer type. So in this case, let's look. Prostate tumors, right, can spread to brain, lungs, liver, and bone marrow. Prostate cancer cells, right, can spread to brain, lungs, liver, and bone marrow. But if you look at the way they've drawn this cartoon, right, the size of the arrow indicates where we typically see more of prostate metastases take place. In other words, the primary site of prostate metastases is bone marrow. Right? You do see some in the liver and the brain, but in the lungs, it's a little bit less. Look at pancreatic cancer. Pancreatic cancer is very different, right? Pancreatic cancer very rarely metastasizes to the brain or bone marrow, right? It can metastasize to the lung, but pancreatic cancer cells, they love to metastasize to the liver. Right? Colon, likewise, also loves metastasizing to the liver. Breast typically metastasizes to lung or bone marrow, also can metastasize to brain, but not typically found metastasizing there, right? So this reminds you about the seed and soil combination, but it makes you also wonder, right? What is it about the different types of cancer cells that make them only compatible with certain types of soil environments? And again, right, this all has to do with the fact that the original healthy original cell, normal cell that gave rise to the cancer, right, already was dependent on certain cellular pathways for being a prostate cell versus a breast or colon or pancreas cell, right? That might be very different than other normal cells somewhere else in the body. And that sort of dependence upon those pathways still might play a very important role in whether those cells, if they get some other tissue in the body and revert back to epithelial, right, they need or expect to be able to still use those pathways. So let's look at bone marrow, because bone marrow is a common place of metastases. Why is bone marrow such a good soil for metastases? It's because the nature of bone marrow is that it is an environment that is constantly being turned over and replenished. So in your bone, so on the left here, you can see that your bone is constantly being reabsorbed, right, to be able to be regrown. Every day, every single day, just from walking around on our 2 feet, the bones in our feet get thousands and thousands of teeny tiny little fractures in them, right? And you might think, what? You're fracturing your bones every single day? I'm not talking about big fractures. I'm talking about teeny tiny microscopic cracks that just happen because, hey, you're jumping up and down walking on your feet. Your entire body weight is on your feet. Of course, the bone, right? Just like the concrete of a bridge, right? When a bridge, if there's some big catastrophe and a bridge collapses all of a sudden one day, right? It's usually not because just that one day something happened to cause it to crack and fall. It's because of the buildup over many decades of teeny tiny cracks, right, that are part of stress fractures. And then one day it's just can't stand up anymore, right? So our bones like that as well. That means every day our bone is being sort of reabsorbed so that fresh new bone can be grown to repair these fractures so that our bones don't just suddenly break one day, right? So that's the job of osteoclasts. So osteoclasts are cells that digest bone for reabsorption. So in this slide here, these big purple cells are osteoclasts, right? And they're digesting away the mineralized bone, right, to kind of rejuvenate it. Osteoblasts are the cells that lay down the newly formed mineralized material. Right? Not surprisingly, if this process is constantly going on in your bodies, right, the extracellular matrix is very rich in this environment with all kinds of growth factors. Right? And so you can see here is an osteoclast digesting away this mineralized bone layer. And you can see in this spot here, it's digested away this layer. And what do you see underneath? This is the nutrient-rich and mitogenic signal-rich extracellular matrix, right? So that these factors can then be released to encourage things like osteoblasts to be able to move around in here and lay down the new mineralized bone. Right? So metastases to bone then takes advantage of this rich environment of mitogenic signaling to grow in. But here, this is the last slide for today, right, is a great example, again, how different seed and soil combinations can lead to very different phenotypes and expression of different tumor types. So here's normal bone, the first pill on the left, right? So this is some skeletal muscle on the right. Here is the bone. You can see here, the orange parts are the mineralized bone, the healthy bone, and in between are pockets of the bone marrow. Right? Here is breast cancer that has metastasized to the bone. And you can see what has happened. Much of the orange mineralized bone has been eaten away, right, and is replaced by the tumor. So we call this an osteolytic metastases, right, because it's caused the bone, the mineralized bone, to be digested away. That is why one of the very important clinical symptoms of breast cancer and metastases to bone is frail bones that are easily broken. Right? Prostate cancer, when it metastasizes to bone, it does the opposite. Right? It has an osteoblastic metastatic state where you can see it actually stimulates far too much mineralization of the bone. Right? And this is the tumor tissue surrounding it. Right? So again, makes you think about, depending on what seed we're talking about and the genotype of that seed and what that seed is programmed to do, based upon what it originally was programmed to do when it was a healthy breast cell or a healthy prostate cell, right? Those types of unique characteristics get carried with it into the new soil environment. And in this case, both breast cancer and prostate cancer happily metastasize to bone because the combination of different stromal signals is right. But this seed-soil combination results in very different types of clinical presentation of disease. All right. Okay, let's go ahead and stop there.